Welcome to the Explainer. Today, we are diving into the rebels and outliers of the genetic world. The biological phenomena that just flat out refuse to play by the traditional rules of inheritance. It's going to be an incredible journey taking us all the way from the petals of garden flowers right down to the microscopic sugar molecules sitting on your red blood cells. Let's get into it. So, for a long time, our understanding of inheritance was basically governed by Gregor Mendel's classic rules. You already know the basics, right? A dominant trait masks a recessive trait. Cross a tall pea plant with a short one, and boom, the offspring are tall. The dominant factor wins. But what happens when genetics doesn't follow those rules? What happens when neither trait truly dominates, or when they both decide to take the wheel at the exact same time? Well, that is the exact mystery we are unpacking today. Here's our roadmap for today. We'll start with beyond basic dominance, then look at incomplete dominance in snapdragons, uncover the molecular basis of dominance, explore codominance and blood types, and finally wrap up with understanding multiple alleles.
Part 1. Beyond Basic Dominance.
Moving right along to Part 2. Incomplete dominance in snapdragons. So, while Mendel's work with pea plants laid this absolutely incredible foundation for genetics, scientists pretty quickly realized that nature actually loves to break its own rules. Let me introduce you to our first genetic rule breaker, the dog flower, commonly known as the snapdragon. Now, if we strictly followed Mendel's rules, crossing a true breeding red-flowered plant with a true breeding white one should give us offspring that are entirely red, right? Assuming red is dominant. But take a look at what actually happens in the F1 generation here. They are completely pink. The trait didn't resemble either parent directly. It just completely blended into this totally in-between phenotype. Let's look at how the genetics actually play out here. We start with our true breeding parents. The red flower has two capital R alleles, making its genotype RR. The white flower has two lowercase r alleles, so RR. When they cross, every single offspring in that F1 generation gets one big R and one little r. But, and here's the catch, because that big R is not completely dominant over the little r, the red pigment just isn't fully expressed. And that is exactly what gives us that signature pink color. Now, let's see what happens when we take those pink RR flowers and self-pollinate them to get the next generation, the F2 generation. And this brings us to the absolute crux of incomplete dominance. When we break down that F2 generation, the offspring come out as 25% red with the RR genotype, 50% pink with the RR genotype, and 25% white with the RR genotype. Now, genotypically, this is a 1 to 2 to 1 ratio, which perfectly matches standard Mendelian rules. But phenotypically, the actual physical colors resulting from those genes, that ratio has totally changed from the classic 3 to 1 dominant recessive ratio. Instead of 3 reds and 1 white, we get 1 red, 2 pinks, and 1 white. The R simply wasn't completely dominant over the R, allowing that pink heterozygote to stand out as completely distinguishable from both of its true breeding parents. Section 3, Molecular Basis of Dominance. Okay, so we've seen this blending happen on a macroscopic level, right? We can literally see it in the flower petals. But what exactly is dominance at the microscopic level? Like, why are some alleles dominant and others recessive in the first place? To tackle this, we kind of have to rethink what a gene actually does inside a living organism. Basically, every gene contains the information to express a particular trait. The absolute easiest way to think about this is to view a gene as a recipe for an enzyme. In a diploid organism, like us, you have 2 copies of each gene, one from each parent. Let's assume the normal, unmodified allele produces a perfectly functioning normal enzyme that's needed to transform a specific substrate inside the cell. When the enzyme does its job, boom, you get the normal trait. So the crucial point is what happens when that recipe gets changed or modified. A modified allele could theoretically produce one of 3 things. First, it could produce a normal, or maybe just slightly less efficient enzyme. In this case, the trait remains mostly the same, acting just like the unmodified one. No big deal. Second, it could produce a completely non-functional enzyme. Or third, it could produce no enzyme at all. In those last 2 scenarios, the trait is visibly affected because the chemical reaction just doesn't happen. The functioning, unmodified allele that produces the original phenotype is what we call dominant, while the modified, non-functioning allele is recessive. Honestly, you see a recessive trait simply because an enzyme is broken or just completely missing from the kitchen. Part 4. Codominance and blood types. Let's move away from plants and into human biology to see how this builds on our cellular understanding. While incomplete dominance gave us that blending effect, codominance does something entirely different. Human red blood cells are actually fascinating. The plasma membrane of these cells has these tiny sugar polymers protruding directly from the surface. And the specific kind of sugar that sits on the outside of your blood cells is controlled by a specific gene, which geneticists call gene I.
And here is where it gets really interesting. Gene I doesn't just have two variations, it actually has three distinct alleles floating around in the human population. The allele IA produces an A-type sugar. The allele IB produces a slightly different B-type sugar. And the lowercase I allele, well, it doesn't produce any sugar at all. Because humans are diploid, each person only carries two of these three alleles. Now, both IA and IB are completely dominant over that lowercase I. So if you have an IA and an I, your body just makes A sugars. But what happens if you inherit an IA from one parent and an IB from the other?
Out of the 6 possible genetic combinations that result in our 4 distinct blood types, let's focus on what happens when a person has that IA and IB genotype. Something amazing happens. They don't blend, and one doesn't mask the other. Instead, they exhibit codominance. Both alleles fully express themselves. This means the person's red blood cells are coated in both A-type sugars and B-type sugars at the exact same time. It's literally a perfect molecular tie, resulting in the AB blood type.
Section 5. Understanding multiple alleles.
The ABO blood group acts as a perfect bridge to understand this concept of multiple alleles. It is incredibly important to remember this distinction. An individual organism can only carry 2 alleles for a given gene. Why? Simply because we only get 1 chromosome from each parent. Because of that, you can only observe multiple alleles, meaning more than 2 variations of a gene governing the same character, when you study an entire population. So in the wider human population, 3 alleles exist for this blood type gene, even though you personally only possess 2 of them. Let's summarize these 2 incredible rule breakers side by side to really lock this in. On one hand, we have incomplete dominance. This is where traits blend into an entirely new in-between phenotype. Just think of the pink snapdragon flower as your mental anchor here. Neither the red nor the white was strong enough to completely take over. On the other hand, we have codominance. This is where both parental traits are fully and equally expressed at the exact same time, with 0 blending. The AB blood type is your anchor here, where both A and B sugars are probably displayed on the blood cell surface right side by side.
Wow, we've traveled all the way from the garden with Mendel's peas and snapdragons right down to the microscopic enzymes and sugar polymers on human blood cells. We've seen that the rules of genetics are really just descriptions of how different protein recipes interact in real time. So I want to leave you with this provocative thought. If dominance is really just a matter of whether an enzyme is functional or not, and if populations carry a vast array of multiple alleles hidden in plain sight, what other basic biological rules do you think are out there just waiting to be broken? Thank you so much for joining me on this explainer. Keep asking questions and keep exploring the incredible code that writes our world.